right? We're going to walk through it. So the document to work with is in Module 9, the workshop notes. Make sure you download it to your computer. Open it, and just to give you a little bit of context, what are we going to do? We're going to assembling the genome is it can be quite a complicated task, and it can require a lot of resources. So what are we going to do? We're going to use a small subset of reads that we'll download from the internet that will allow us to assemble the genome of chloroplast. So chloroplasts, they're photosynthetic organelles. They're located within the cells of mainly plants and algae, green plants and algae. And they actually have their own genome. So the theory was that at some point they were standalone prokaryotes that at some point became symbionts with plants and then became embedded in them. But still they have their own genome apart from whatever whatever species they are inside. So this is an example of the tobacco chloroplast genome. So it's like a prokaryote, like a little bit like bacteria. It has a circular genome. And you can see here the genes on the genome. And the size of the genome in the tobacco is roughly 156,000 base pair. So remember that sort of structure and number. And what we're going to do, we're going to assemble the chloroplast genome of the sweet potato. And then we can compare it to the one that we have here of the tobacco and see how good we did. All right, so the first thing we want to do is we want to log into Galaxy. And then I'm going to give you a bit of explanation about Galaxy. So to log into Galaxy, first go into the usegalaxy.au. usegalaxy.org.au. First go to the Galaxy website. And then if you haven't logged in, for my case it's already logged in. If you haven't logged in, click on the user and then it should say, instead of user, it should say login or register. Oh. And then when you do, wait a little bit and there's going to be this orange login using AAF. So that will let you log in using your Griffith credential. I'll show you how it looks like. If I log out, then you've got the login or register here, and then if you click on it, it will take you here. Rather than trying to put a username and password, just click on the sign in with AAF. The orange button. This is like a federated login for Australian academics, and then it will take you, probably if it's your first time, it will ask you which university you are and just choose Griffith. And then it will take you to the Griffith federated login where you provide your username and password. All right, if you have any trouble at any point, just raise your hand, and either myself or Nikki will come and give you a hand. All right. Alright, once you've logged into Galaxy, go to the workshop, and you'll see that there's a link here. It says go to Galaxy Australia with a longer link. So follow that link. It should say usegalaxy.org.au/join-training/genome-assembly-GU. So follow that link. And it should show you this one. Congratulations, you're successfully registered in Genome Assembly GU. Yeah, you haven't logged in yet, I reckon. First login you can do with Galaxy. Yeah, you are logged in. Because it opened it in a different browser. Can you just copy this? And you used Chrome before, right? Yeah, now put it in there. Alright, now return to Galaxy. Alright, so again, first log into Galaxy, and then after that follow that link. Yep, great. Alright? You need to put it up there, not in search bar, you need to put it in the Alright, basically what that means, you see now it says Galaxy Australia Training Infrastructure as a Service. It's actually a specific Galaxy with resources available for us for this workshop. So it makes sure that our jobs get processed quicker and we don't get stuck in the queue. Can you download the new? No, that's variant analysis. Everyone all right? All right. Now, once you got here, just click on Return to Galaxy, and it will send you back to Galaxy. But again, this time we will have we can use sort of, again, special queues. Our jobs will be processed quicker. I hope. Just let me close the other Galaxy that I have in here, otherwise I'll get confused. All right. Now, let me explain you a little bit about Galaxy and how it looks like. All right. So just to start with, what is Galaxy? I told you a little bit just briefly, but Galaxy is a web-based analysis and workflow platform. And basically it's an interface to quite powerful computing cluster that runs Unix in the background or Linux in the background. So the advantage of Galaxy is that it gives us the graphical user interface to run all these jobs that otherwise we had to do them in Linux or in the Jupyter. There's other advantage as well when we talk about workflow, and we'll see about that it makes it much easier to link between the input or the output of one tool to the input of another tool. So it makes it much easier to work our procedures and our steps in the workflow. It is often hosted on national, state, or institutional infrastructure. In our case, it's actually national. So the Galaxy Australia is serving all of Australian users. It's been hosted by UQ, QCIF, and in Queensland, and also developed by Melbourne Bioinformatics. It is designed for biologists, and so it really has a lot of the tools, the very common tools that we use in bioinformatics. As you can see, it's very easily accessible. We just need a web browser. It doesn't matter how powerful our computer is. All right, the main thing there is all the computation is done in the background, and the web is only showing us the results. We can upload our own data, or we can access shared data, and that's what we're going to do in the workshop. As I said, it uses very common bioinformatics tool, with well-cited, well-respected. So if you use the tools in Galaxy, you know that no one can tell you something about why you use that tool or the other. We can develop very customized workflow. And Nikki is a good example. We're working on some of her data, so really specific to the exact experimental design that you have. And there's really great knowledge base of manuals and training materials. And often they run workshops as well. So QCIF, which is a Queensland Computing Infrastructure Facility, they actually run workshops on how to do certain things in Galaxy. And you've got like a full day of working in Galaxy. And we're going to see that training material is really great. So we're going to follow in some, partly we can follow some of those available tutorials. And I'm going to show you them. All right, so the layout of Galaxy. The left side is the available tools. There's a search bar at the top, sorry. At the moment, if you'll have a look, it's separated by sort of tasks. So you've got genome assembly, you've got variation, you've got transcriptomics. But if you start typing the name of the tool, it will filter it down and it will show up the tool that matches your search result. The middle one is the main sort of view. When you just log in, it's like the home page, so it's got news, updates, and so on. Once we start, when you click on a certain tool, all the parameters, all the options of the tool will show up in that middle one. And when we look at results, that's where we're going to show up as well. So that's where most of the work happens. On the right-hand side, we've got our history. So the history contains all the data that we uploaded and processed, and all processes, so we can keep track how they're doing. And we'll see how it works as well. We can have multiple histories. So let's say this workshop, we're going to have one history, and we call it genome assembly. Next week, we're going to have another history called genomic variation, and they can live side by side. So as long as you don't reach your quota of storage, you can just keep your history side by side, and you can always transfer files between one and another and move between them. At the top, we've got the menu. And the analyze data, this is sort of the home page. I think in our case, it doesn't say analyze data, there's just a home icon. Workflow is where we can design those workflow to automate processes. Shared data is where we can access shared data, shared workflows, and so on. And the help user is where we can set user preferences, and the help is where we can access all the training material. Again, in Use Galaxy, the help one will just have like an academic hat icon on it. Galaxy, specifically for Galaxy Australia, I mentioned who hosts it, and as I said, there's specific training materials that were developed right here. You can access it pretty much from every modern web browser, all right? So you can even do it on tablet and so on. What we're going to run the genome assembly tutorial, as I mentioned. It runs on a dedicated Galaxy queue, so that training infrastructure, again, what it means that there's a subset of computing resources that are set only for us for this workshop. So hopefully things will run quicker. If you want to repeat that, if you want to go back to it again, that's in the lecture slides as well, that link. Now, you see it says follow the steps at the chloroplast genome assembly. And I think we should have those links in the workshop as well. Yep. So if you see here, open the chloroplast genome assembly tutorial. And so you can open up in a new page. And this is an example of a tutorial, of a Galaxy tutorial. All right, it's very nicely put together. In an HTML format, so it's a web page. You've got the table of content on the left hand side that you can scroll through, and often there's good introduction. There's the content, and then when we'll go through what we're going to do most of the hands-on sections. But again, we're going to make some slightly differences. But overall, we're following this tutorial. So the first thing we want to do is create a new history. Right? By default, when you just logged in to Galaxy, if you didn't have any previous history, like most of you, on the right-hand side, you'll see an empty history. So you just say unnamed history. Right? If you previously worked with Galaxy, you might have there something previous, and then you need to create a new one. The first thing we want to do is name that history. Right? And then later on, when we want to get back to it, it makes it easy for us. So you see this pen icon right next to the history. So if you click the pen icon, it lets you edit the name of the history. And let's call it chloroplast genome assembly. Right, you can provide additional description at the bottom if you like to, for your purposes. I'm going to leave it at it for now. But yeah, you can write there, tutorial in applied bioinformatics course or whatever. And then you save it. Sure, I have saved it. I think it didn't save it for me because I had the same name before. All right, give it a name. So now we can get back to it whenever we want. That's the first step. Again, if anyone's stuck at any point, just raise a hand and we can help you out. Now the second thing we want to do, we want to upload our data in there. So let's have a look at the tutorial. And what we're going to upload two sets of reads. A FASTQ file with Illumina reads. That's called sweet potato chloroplast Illumina reduced, and a FASTQ file with nanopore reads. It says sweet potato chloroplast nanopore reduced files. Now it's quite easy, and you see both files are hosted online on a website called Zenodo. So what we're going to do, we're going to copy. So you see in the black bit, you can either highlight or there's this copy button. So you click the copy button, and it gets copied to your clipboard. And then we go to Galaxy, and you see on the left-hand side where the tools are, the first one is upload data. So click on upload data, and then at the bottom one, choose paste/fetch data. All right, that gives us this dialog, and what you need to do is just paste what we copied in that text box. And once we've done it, we click on start. And then you'll see what happens when Galaxy is doing things. So process this request. You can place close after that. Then wait a little bit, and you'll see on the right-hand side. You might need to click refresh if it doesn't refresh automatically. Yeah, you click on start. Can you have it? Yeah. Yeah, it might take a bit of time. To do its thing. Once you click on start, it should think a little bit, but then it should do this status. Now what you should see on once this managed to submit the job, you should see here 2 gray boxes. Oh, there you go. It turned with green. Should have touched it. So usually it starts as gray. Gray means the jobs has been submitted to the queue. Then it turns orange, means they're being processed. Then they turn green, means it finished processing. All right, so these are sort of the steps. And once it's finished, once we've got those data sets in green, it means that they have been uploaded. And then we can briefly have a look at what these files contain. If we just click on the file name, and it will open this little dialog box saying the size of the file. So you see this file, the nanopore file is 25 megabases. The format is FastQ Sanger. And even though it says Sanger, no, it wasn't produced by Sanger sequencing, it's just the FastQ format. And it shows us just a little bit, just like the first 5 lines of that file. Now it says the format is FastQ Sanger. Do you remember, have you watched the lecture, what is a FastQ format? All right, so let's do that. Let's click on the, now if we want to see the content of an object in our history, we can click on the eye icon next to it. So let's click on the eye next to the Illumina dataset. And again, if in your case it's still orange, just give it a bit of time. It will finish uploading. All right, and it gives us the content, and it tells us that this data set is large, and only the first megabyte is shown, right? Because it just gives us sort of a preview. And this is a typical FastQ format. So each record has 4 lines, starting with the @ sign. So as opposed to regular FASTA, which starts with a greater than sign, here records start with the @ sign. And we've got the information. I told you I was mentioning in the lecture that often in FastQ file we've got the header will contain information about the flow cell, the sequencing platform, and so on, the barcode, the indices. In this case, it says SRR something, something, something. The reason is because these were downloaded from the NCBI short read archive. So it just gives us that catalog, that NCBI accession number. If we want to know all the details about this particular sequencing, we can go and search for that on NCBI and we'll get all the information. Then we get the actual sequences. So this is the actual read that was produced. The third one is always just a plus. And then the 4th line is the quality of each of those bases. So A got a quality of J, T got a quality of J, and so on and on and on. Those quality, as they appear like that, they're very hard for us to interpret. They're pretty much code for Phred scores, for quality scores. And looking at it just like that, it's not very useful. We'll see how we can process it with a tool that gives us summary statistics of the overall quality of our reads. Now, one thing to have a look and recognize is the length of the reads. All right? What can you tell about the length of the reads? Are they varying or are they fairly constant? They're fairly constant, all right? So this is very, very sort of a typical characteristics of Illumina short read sequencing. Most of the reads will be the same size, the same length. Now if we have a look on the nanopore one, so I'll click on the eye next to the nanopore sequences. So you'll see a very, very different very different view. So if I'll just scroll to the right, you see that the reads are much longer. And you see that they're in varying sizes, all right? This one, the first one ends here, the second one actually continues past it. And you can see, again, very typical nanopore or PacBio sequences. Much longer reads, but varying length. Illumina is always the same, or the same length. All right. So this is just like having a peek into our reads, just to get a feel of how they look like. Now what we want to do, we want to assess the quality of the reads. And to do that, we will use several tools. The first tool is called FastQC. So put the cursor in the search bar of the tools on the left-hand side and type FastQC. Now wait a little bit. See the thing is loading, and it will filter the tools. You can actually access it directly from the FastQ quality control, but it's easier to just type the name of the tool if you know it. Yeah, there you go. Now it shows you two tools, FastQC and FastP. We're actually going to run both, but let's start with FastQC. Read quality reports. Now that's where Galaxy is so easy to use. So when you open FastQC, FastQC in fact is a Linux-based command line tool that you need to provide it with arguments and which files to operate on and so on. In Galaxy, when you click on a tool, it will open the tool options and inputs. Mine is a bit slowish. And you'll see that the other advantage of Galaxy that it will only, as the input files, it will only show you input files that are compatible with that tool, with the input for that tool. So FastQC by default takes as input FastQ files. Oh, come on. Jeff, it's my computer. Oh, there you go. All right. So you'll see, but by default, if you search for input files, so you see the first argument that you need to provide is the raw read data from your current history. And if you click on it, you'll see that the only options it shows you is only your FastQ files. Now it's easy because that's the only files we got in there. All right? But even if we had more files, it will only show FastQ files. But what we want to do, we want to run it on both of them. So if you click the middle button here says multiple datasets. You can choose both of them. You need to press Control and click on both of them. And if you have a look at the bottom, it says this is batch mode input field. Separate jobs will be triggered for each dataset selection. So we can provide multiple datasets, but it will run FastQC on each one of them. So instead of running it twice. All right, so we choose the middle one, multiple datasets, select both datasets, and we can leave everything else as default. We can provide a lot of extra information for FastQC, but the default should work great. Now just have a look that if you scroll down at the bottom of the tool, you've got a lot of extra information, which is very useful. About the inputs and outputs, what sort of output is provided, the citation for the tool, and so on. So once we specify the inputs, we click on Execute. Execute will send the jobs to our history. Again, mine is a bit slowish, but you're supposed to get those jobs in gray. Right? All right, but you chose those. All right, be patient with it. It takes a bit of time until it loads and submits jobs, but scroll down to the bottom.
I think you see like very small.
You need to choose both.
I need to hear the recording.
Just press control.
I need to hear what the recording
Just
is.
press anyone. One of those. All right.
Hold. Oh no, upload separately. So I'll upload it before we got no, it's 7 is old. Please read it.
Yeah, and then scroll down. Click exit.
Read it.
Yeah, press control. Choose the other one as well.
It's actually
Yeah,
confusing.
scroll down. Click exit.
Like, Frank didn't go through the well.
All right. So you see, it's very quick.
How I did.
So by the time it refreshed, they turned green as well. So it's already finished. And what we're gonna do now, we're gonna do the same thing, but we're gonna use a tool that's called FastP. All right, just a minute. I'll show you an example of the output of FastQC. Nikki, I think they might need some help over there. Now, all right, can you pay attention, please? So we've got from each. Now this is where it's really worth renaming the output that we got, because at the moment, what it says to us, it says FastQC on data 2 and FastQC on data 1. And then we need to start scrolling back and say, well, what was data 1, what was data 2? All right, and it can get a bit confusing. So what we're going to do, we're going to edit with the pen, and instead of saying FastQ on data 2, we can say FastQ on nanopore reads. And I will save it. And the same thing for the web page below. So instead of data 2, I'll just replace it with nanopore reads. And then the same thing for the FastQ on data 1. I still couldn't find a nice way to do it automatically. So on that, instead of data 1, I will say Illumina reads. Instead of data 2, I will say nanopore reads. All right, and then when I look at it, it's very clear to me which one is data 1, which one is data 2. All right. Now let's have a look. Let's have a look at the FastQ on Illumina reads, and click on the web page, not on the raw data. Click on the web page output, and on the eye next to it. And basically what it does, it shows me the FastQ output. So again, it's an HTML web page. It's got these basic statistics. So it's got a summary, and it's I can click and it will take me to the right section, and we can start getting some information. So this was the file name. The number of total sequences, 62,500. All right, so this is something we actually want to look at. So let's go to, all right. So in the workshop, you right? All right, I'll come and have a look. All right. All right. Now what we can do, we can fill this table for nanopore reads and Illumina reads based on the FastQC report. All right. So the first thing is number of reads. So I can go to my FastQC report. Just keep track on which data set you're looking at. So in the case of Illumina, the total sequences is 62,500. I can paste it in here. For say that. GC content, we can as well pull it from here. 38. The read length range will be here, 101 to 127. And what else we after? All right, now before I'm going to look into other, I just want to show you the distribution of small thing, but in terms of the alright, something I want to show you, and something that's always we need to consider when we look at quality of reads. So this is the distribution of the Phred score qualities at each position of our reads for all of our reads. Alright, so the distribution, it's a box plot, and it shows me the bottom range and the top range, and the quantiles when we do have them. So what we can see that these reads, they've got pretty good quality all the way up to, I don't know, 80 base pair or something, 75. And then there's some outliers. And again, the majority of the reads where the yellow bars are still over 30. But as we get to 115, 110, it starts dropping below. So some reads, some base qualities are dropping below. And we can see as the longer the read gets, like the end of the read, dropping in quality. All right. What it tells us as well that not all of our reads are top quality, or not all of the bases in our reads. So we need to do some quality trimming to remove some low quality bases, and I'm going to show you how we do that. Now in terms of there was a question there about the mean quality scores range. Quality scores range. I think we can pull that one from a different file, so just leave that one empty for now. Yeah, another thing that FastQC will tell you is adapter content. So often when we get the data from the sequencing, it has all sorts of adapters that Illumina that were added during the library stage preparation, and they're not part of our actual sequences. In this case, the adapters were removed. But if you look at raw sequencing data, you will have all sorts of adapters in here, which tells us as well that we need to remove them. That's pretty much it. The GC content allows us to identify contaminations. So sometimes you'll get 2 peaks here, which means that we've got DNA from 2 different sources, because each genome will have its own pattern of GC content. You know what is GC content? All right, it's how much G's and C's are in our DNA. As simple as that. All right. So and it represented as a fraction from 0 to 100. Just a percentage of G's and C's in our genome. And each genome has a very unique pattern. So if we've got contamination, we'll see 2 peaks in there rather than just one distribution. square-based sequence content, it's all right. And again, when we look at the sequence quality, we see that the vast majority, so this is distribution, so the majority of our sequences are around 30, 39 Fred score, which is really good, but we've got some, actually this shows us the mean quality range, so it ranges from 15 to 41, pretty much. So we can use this information to fill in the mean quality score range. So we can say it's 15 to 41. All right. Now let's look the same thing for the nanopore data. And please go ahead and copy those inputs for the nanopore data. So have a look at the range of different, and we'll have a look at the distribution of length. Qualities, have a look at the qualities. Much worse. All right, all of them in the red, except just a few outliers up there that in the yellow zone. But overall, the beginning of the reads are really poor quality, and then the majority of the read, like the majority of the length of the read is very average, if not poor quality. Maximum of 20. Definitely nothing impressive. Saying that, this is probably resulting from very early generation nanopore data. Nowadays, nanopore data is much more accurate, in much better quality. Per sequence quality scores, you see there's a much larger distribution, and we can see that it starts anywhere from, we can put the range of 5 to 18 as our quality range. And the sequence length distribution, we can see, is much more varied than the Illumina one. In the Illumina one, we had almost, the reads were almost all of them in the same length. Right? And as well, the GC content is very biased just because we only have 2,000 reads. There's just not enough reads to produce a good distribution. Right? I provided a link for an information about how to interpret the result of FastQC. And there's a question there based on the table above. Briefly summarize the main differences between the reads from the two sequencing platforms. It's quite obvious. You just compare each of the values one to one another. And you can, it's quite, the differences are quite obvious. All right, now what we want to do, we want to trim low quality bases and adapters and so on from those reads. So we're going to use a tool that's called FastP, and it should already be on your left hand side if you didn't remove FastQC. See the second tool there is called FastP. So we can click on it. And let's put it as well in batch mode. So let's leave it as single end. Choose multiple data sets, and then choose both data sets with control. Now there's one thing that we want to do here that's different from the default. In the output options, I'll just zoom in a bit. In the output options at the bottom, open it up and choose output JSON report as well. change it to yes. All right. So at the bottom in output options, choose output JSON report, make it a yes as well. And then click execute. So very similar to the FastQC, but now we apply FastP. What FastP will do, it will also generate quality measures like FastQC did, but it's also going to trim down any low quality bases. All right, fantastic. Great. Now what we want to do, we want to assemble our genome. So we got the raw reads, we checked their quality, assessed their quality, did adaptive trimming, and now we can assemble the actual genome. Let's, in the tools, search for a tool called Unicycler. And you'll find something that says create assemblies with Unicycler. Now, before we put any input in there, I want to scroll down to the bottom, and you can have a read a little bit about the options and what is Unicycler. So Unicycler is a hybrid assembly pipeline. Can anyone recall what is a hybrid assembly? Most of you weren't yesterday morning, so you probably don't know what I'm talking about. But if you've seen the recording, all right, a hybrid assembly, it means that I use a combination of long reads to have the scaffolds and the continuity, and then I use short read, Illumina short read in between to get better accuracy for those bases for that assembly. So there's two ways to do that. We can either do first an assembly with long reads and then after that lay on top of it and what we call polish the assembly using the accurate Illumina data. The other alternative is to straight away use a tool that can use both types of reads, and that's what we're going to do with Unicycler. So it's a hybrid assembly pipeline for bacterial genomes. It's very important. So we shouldn't try to use that for a plant genome or any eukaryote. And as I said, it uses both Illumina and long read to produce complete and accurate assembly. Now let's scroll up. And it gives us information about the input data and what is the bridging mode and so on. All right, so let's scroll to the top. Whether my data is paired end or single end, our data is single end. Paired end is when we have forward and reverse reads for the same insert. So let's change it into single. Select unpaired reads. And what we want to choose is the FastP on data 1. All right, again, it would have been great to rename it, but I think I remember that data 1 is Illumina and data 2 is nanopore. So choose FastP on data 1. So these are the Illumina reads that has been quality trimmed by FastP. And then select long reads. Let's select FastP on data 2. Bridging mode. Let's make it bold. Longest config, higher misassembly rate. So trying to get as complete as possible, even if there will be some inaccuracies in there. The expected number of linear non-sequential sequences in the assembly is 0. Yeah, let's leave all the other options just as is. And let's click execute. Have you submitted it? No. Oh yeah. Yeah, no.
And then right after the body quiz, they were like, trash. Which one? Oh. Yeah, that's what he sees as well. Yeah.
And it might be a good time now to rename our previous FastP outputs, again to replace data 2 with nanopore and data 1 with Illumina. So do it for the JSON report. Well, you can do it for all of them. So the FastP on data 2, just replace data 2 with nanopore reads. Probably would have been better to do it before we ran Unicycler, but that's all right. Just to help us identify which one is which. Just when you do that, be careful. Well, if by chance you press on the delete icon, which is right next to the edit icon, alright? Don't worry, it doesn't disappear. It just goes on the top here. You can view. At the moment, it only shows us the active datasets, but deleted datasets will be there as well. All right, so if you deleted something by accident, we can always retrieve it back. Yep, and then I replace data 1 with Illumina reads. Must be something that say like rename and just replace data 2 with or just replace all of them. All right, great. Mine finished, so I hope yours will finish soon.
What? Oh, you're talking to me?
Yeah.
I thought you were talking to yourself. Yeah. Yeah, I wasn't sleeping. Week 8 was no use watching Friday. I mean, you can watch it. Because he said like last week he used a different example. Yeah. So I'm like, what is Frank trying to say?
All right, and while we're waiting for this, I want to show you then how we assess and how we visualize sort of the assembly that we got. So in the tools, search for bandage. And bandage, what it does is visualize the Nova assembly graphs. Now before we run bandage, scroll down to the bottom just to see. So there's a little bit of description, what to expect from bandage. And they've got some examples. So if you have a look at the examples, this is a perfect sort of bacterial genome assembly. We've got a single circular genome. All right, this is everything we expect. This is sort of the best possible option. Right next to it is a very poor assembly. So all the different colors mean different contigs. So they couldn't get a nice continuous assembly, and we've got very small fragments, and it does its best to put them together in some meaningful way. But as you can see here, we've got a lot of sort of loops, and we've got dead ends, and it looks like just a one big mess. So pretty much there's not enough overlap to be able to determine the correct structure of the genome. So that's not a great output. If we scroll a little bit to the right, we see if we look at particular regions, it shows us a particular vector that the graph shows something like that, and our interpretation can be that we've got like a circular vector and then a linear gene next to it. So that's which incorporates the same feature in it. All right, so several ways that we can sort of interpret the output. Let's see what we got. So let's go to the bottom of this page of bandage. And the input that we want to provide. Yeah, even if it's still loading, now this is the nice thing about Galaxy. We can set the next tool, and it will wait for the previous tool to finish before it will send us the next job. So we can set the jobs, future jobs, and only once those outputs are available, it's going to start processing. So nothing stops us from going to the next step. So it asks us which assembly we want to provide. So we can choose either the actual FASTA or the final assembly graph. Let's choose the final assembly graph that we got. Image height, let's leave it as that. Image width. Node name labels, click on yes. Font size, let's put 20. And produce, what type of file to produce, just leave it as JPG. And click on execute. Now if it already finished, like in my case, it will go straight away with bandage. If in your case it's still loading, it will just make it gray. It will sit in the queue until the output of unicycler is done, and then it's going to execute it.
Support short cost. Sure.
Yeah, what you can do,
You mean your laptop doesn't send the answers?
Yeah, I don't think it's saying.
all right.
So like having to redo it.
Oh, there you go. Mine finished. There you go. Alright, this is what I got. I assume that most people got, sometimes it looks a bit different shape or everything, but what we're getting a single strand, alright? A single config. And if you want to, you can right click on it and open it in a new tab. If you just want to admire it a bit more. It might not look impressive to you, but actually it's a damn good result. Basically what it means that it was able to create one single continuous assembly, all right, rather than multiple contigs. The size of it, can you read the size of it? It's written there, it's a bit small, we probably should have made a bit larger. 130, mine is 130,069. Yeah. Yeah. Again, there might be slight differences.
What does this number represent?
The length of it. How many base pair? Right? How many base pair in that genome? Alright? So yeah, what do you think? Alright, now let's first of all I'll copy it and I'll paste it. I'll copy the image and I'll paste it in here.
Excuse me?
Yeah.
Why is it showing one?
No, the one is the number, but underneath the one, there should be the length of it, and if not, it's because you didn't specify to get the.
One 30069.
But we can look at it as well. If you look here in the final assembly. Alright. Yeah, well, they come up in different shapes, but they're the same, sort of the same principle. Now, let me ask you a question. So if you scroll up to the very top of the workshop, of the tutorial document, and you know what? Before we do that, let's figure out what's the length of it. Again, you can read it somehow from here, but if you just click on the final assembly, the first line of it will tell you the length. Probably should have asked as well, what is the length of the genome that we got? I'll just paste it here. Alright, and there might be slight variation. Maybe your genome came up as 130,061 or something like that, but they're all fairly similar. Alright, so scroll to the top, have a look at that tobacco genome assembly. Tobacco chloroplast genome assembly. If we compare this one to the one that we've got, what do we think? Do we got the whole chromosome or not? Why do you think we didn't?
We didn't complete the circle.
Alright, we didn't get the full circle. Alright, it couldn't complete a circle. Anything else that might suggest that we might be missing something? Yeah. We have less base pair, right? Slightly shorter, right? And we don't know exactly. There are different species, but we we got like 25,000 base pair less than the tobacco genome, and it is not completely circular. Right? So it might indicate that, yeah, we've got one continuous assembly, but it might be missing some bits to have the full circular genome.
Also, because we trim few base pairs, so there's a possibility that those base.
No, well, just because yesterday we didn't trim and we assemble it without trimming them, and it still comes out the same. It's just the data that we have doesn't cover everything. Right? And it's important to notice that we used only a subset, so we only used 2,000 nanopore reads and only 65,000 Illumina reads to produce that. So it's very, very small data set, but still we got a pretty good assembly. Right, so you can discuss this here a little bit. It remains the full sequence of the chloroplast genome. Now part 3, don't worry about part 3. All right, part 3 is only if we assemble the genome with fly and then we polish it later. All right, in this case, we went straight away with unicycler that includes the polishing step in it. All right, so if you want, you can either remove that or you can just say that unicycler included a polishing step and therefore there's no need to perform another.
Oh, therefore it is not necessary to include.
All right, now let me ask you something. What else is missing from our genome? If we look at this genome assembly, what does this genome assembly has that ours doesn't? Annotations. All right, that one has all the genes on it. Ours at the moment has none. All right, we only got one sequence, and we don't know which genes it includes. All right, so the next step, we assemble the genome, we assess its quality. There's more steps that we can run to assess the quality, but we'll leave it for now, unless we'll have more time. The next thing is to annotate the genes and the features on the genome. Again, luckily, we're working with this very simple prokaryote genome, so we can run it very quickly and very easily. When we're dealing with eukaryote genomes, it's much more complicated. We've got splicing, we've got it's much harder to predict those protein coding sequences. In prokaryotes, it's much easier. So what we're going to do, we're going to go back to Galaxy, and we're going to type in the tools Prokka, P-R-O-K-K-A. And as you can see, it tells us Prokka, prokaryotic genome annotation. So click on Prokka. And it tells us which contigs we want to annotate. So yeah, we're going to choose the final assembly. We only have one FASTA file in all of our history. There you go. There's not much we need to change anything. We can change the genus name. Let's call it sweet potato. Again, we should well, you know what? Let's do it properly. Let's do it properly. Let's find the scientific name of sweet potato. There you go. Batatas? Yeah. Ipomia batatas. There you go. So the genus will be Ipomia, and the species name will be patatas. Plasmid name or identifier, let's call it chloroplast. Right? And let's leave everything else as is. So we got our hypomia, patatas, and the plasmid name or identifier is a chloroplast. Just notice that we can change the similarity E-value cutoff. So basically what it will do, it will take pieces of those sequence and it will blast them against known databases to identify the genes. And we can specify the threshold that we want to use for that. Let's just leave it as default. And let's click on execute. You know what? Yeah, let's just click on execute. As you can see, Proca produces a lot of output files. We're only interested in only some of them. Now, while we're waiting for Proca to do its thing, let's align the reads back to the reference to the assembly that we just created. The purpose of that is that we can look exactly how the reads contributed to that assembly, and we can have a look later on and assess if there's any differences, if there's any SNPs, look at the quality, and so on. So let's choose a tool called BWA-min2. BWA-min2. Let's send the proper jobs. Oh, there you go. All right, BWA-min2. Right.
Search for the gene of the interest. I'm like, okay.
Now we need to tell it what is our reference genome that we're going to map the reads against. And it got like, sometimes it's got built-in genome indexes. So for instance, it has already the human genome or the mouse genome and things like that. But we want to use one from our history. Use a genome from history. So we want to use the assembly that we just created. And then the data set that we want to use will be, this is unicycler, the final assembly that came from unicycler. That will be our reference genome. Now the FASTA sequences, now the reads, let's change it into single. And let's choose the FASTP on Illumina reads. Alright, if you didn't rename it, it should be FASTP on the other one, if you haven't renamed it. Leave everything else as is, just double check that the analysis mode says Illumina mode. And click on execute. And let's do the same thing for the nanopore reads. So BWA-min2. use a genome from history and choose our unicycler's final assembly. Change it to single end reads. This time choose FastP on nanopore reads. And now the analysis mode should be nanopore mode. Yeah, nanopore 2D. And then execute. Alright, so again, our FastQ dataset will be FastP on nanopore reads, or trimmed reads and trimmed nanopore reads. And then the analysis mode should be nanopore 2D reads mode. And execute. Yeah, FastQ on data 1. Yeah, that's perfect. You already got to show somewhere. Yep. Now execute. And now do the same thing. Click on the W again. And then change the reference. Change it to single. Change that. FastQ on data 4 on data 2. No. Oh yeah, no, perfect. Yeah. And now change the mode. Alright? Yeah, I think
There is something about me that if let's say I'm busy doing something, like for example studying, I forget about my
Alright? Everyone got the BWA jobs? Alright, now what we want to do, we want to run a tool called QualiMap, which will assess the quality of those alignments. So let's change it into QualiMap. If you search in the tools QualiMap, QualiMap.
Yeah.
Alright? And then choose QualiMap BAM QC, the middle one. Let's choose the multiple datasets in the middle. And choose the two outputs from BWA. You know, before we do that, let's rename those BWA outputs, because I can't really remember which one is which, because now it gets complicated. It says BWA on data 7 and data 15. I'm not sure what data 7 is. So I'll just scroll down. Data 7 will be my Illumina ones. Alright? And number 10 will be my nanopore ones. So I'll just rename BWA meme on Illumina reads, or Illumina FastP, Illumina trimmed reads, and assembly, and final assembly. And then the other BWA meme on data 10, I will edit it I cannot edit it until it finishes. I'll just wait a bit for it to finish. Oh, there you go. Mine finished. So I'll edit it, and instead of data 10, I will call it nanopore trim grids. And final assembly. Alright, so I just rename both, I renamed the outputs of BWA MIM. So I know which one of them refers to the Illumina one, which one of them refers to the nanopore ones. Right? You have to wait until it finishes processing to do that. Right, after I rename them, I can do the quality map stage. So quality map BAM-QC. Choose multiple data sets and choose both nanopore trimmed grids and Illumina trimmed grids that were mapped. You can leave everything else as default and just execute.
So big.
But like more than even Mark, really? The shop. So you stayed away from that vacancy all the way to you're like
very, very old because I have to make it near the waters.
Oh, Sophia. Hey. Let's relax. Somebody survived with 4 hours of sleep.
Yeah. Sometimes like if I have like something important that I have to do, but like it's just I would do it. Like I do it for the 8 hours, like every
All right.
time. And then I'm like,
All right. Now what I want us to do, what we're going to do now, we're going to run a tool called multi-QC. And I want you to pay attention because there's a lot of input we're going to provide to multi-QC. So what multi-QC does, it does QC reports similar to FastQC, an HTML report, but we can provide the output of multiple tools in it. So choose multi-QC. Venom? All right, and we need to say which tool was used to generate the logs. So let's choose Qualimap. All right, let's start with Qualimap, and then provide both Qualimap. Choose both of the Qualimap outputs. Alright, the first one, so the tool that was used was Qualimap, and we choose both the Qualimap. Now click on the plus for insert results. Which tool was used to generate? This time let's make FastP. Alright, and it will let you choose both the JSON reports from FastP. That's why we had to make it JSON, otherwise wouldn't have accepted it. Click the plus for insert more results. And now let's choose FastQC. And insert FastQC output. And let's find those FastQC. Should be fairly at the beginning. There you go. FastQC. Just make sure you choose the raw data, not the web page output. Number 6 and number 4. Right? Now last thing, let's insert more results, the very bottom one. And let's see if you can process Proca results. I'm not sure. Yep, there you go. So choose Proca. And choose the log file from Proca.
The raw data, yes.
At the report title, you can say QC of chloroplast.
Then after the
Assembly.
next one is Proca.
Or chloroplast genome assembly.
Yeah, correct. What is number 2 though? No.
Alright, I'll just, before you press execute, I'm just gonna go over
The log.
the
That's 42. Yeah.
all the outputs, all the inputs that we provide. We provide the results from Proca as the log file, the output of FastQC, the raw data ones, and the outputs of FastP, the JSON reports, and the output of Qualimap, the Qualimap output. Alright, And once we've done all of those, we can click on execute.
Was there
Do we choose the log data for program?
Yes,
He didn't.
there's going to be only one proca. It only worked on one file on the assembly.
Yours says error.
Yeah, so does his.
You can put a file if you want. Do you like it?
How come it says that this is pretty much error? I cannot run this.
Yep, but before you execute, alright. Got a feeling why I didn't like it? Yeah, there you go, that's what I thought. Alright. This is example, alright, of an error. We didn't select the right input file from Proca. I wasn't sure which one of them to use, the text file or the log file. I chose the log, was the wrong one. Now we can easily recreate that, the job, and fix it. So just pay attention. If you click on any of those failed jobs, so just click on the job itself, it tells you an error and it tells you what's the problem. But at the bottom, you see all these icons. So there is this arrow, run job again. So click on it, and it will pull up the job with exactly the same settings that we provided. So what we need to change is in the Proca one, change it into the text file rather than the log file. Alright? So again, go to the error message by clicking on either one of those failed ones. Click on the repeat, run this job again, and then change the Proca to the text file, not the log file. If you haven't submitted the job yet, just do it with the right one. And then you can execute. Right, and once we execute it, we can then later on remove the failed jobs. Leave everything as is, don't change anything else. So choose both of them, yeah. Everything else should be stay the same. Yeah. Alright, everything else should stay the same. Just scroll down to the Proca one. Scroll down to where you find Proca. And change it. Alright, that's good. Text one. Click execute. Yeah, these are not the right ones. Let's see. So this should be scrolled down. Yeah, alright, yeah.
So that's number 41.
The last one.
Let's see. So you go twice, so you can remove this step. Alright, you go faster to see. Now let's choose the right one. Scroll down here. Scroll down. The roll down. Then scroll down to the fast you see. This one. Yes. And it should be, I think, the top one. I think 4 or 6. No, 6. Because I don't know which one is the web page and which one the rollout. I think that's right. Now let's insert another one. Insert results. And fast P. Choose fast P. And then choose these two. Yep, now it's okay. Yeah, you can delete the failed ones. You just missing the fast P. Yeah. No big deal. Missing the fast P.
You what?
You can delete the failed ones. This one. It's alright. No big deal. Yeah, leave it like that. Scroll down.
You're going to cook up
This is both of them and
Scroll down. And then we'll just find your section.
one is for the correction of the dot
Scroll down.
That's what
Yes, so did you do
beef bones.
the option not coming.
This one should be fast paced. And then choose these two. Yeah, no, scroll up. Scroll up. Scroll up one way. Fast paced, let's see. Scroll down. We want focus as well. Scroll down. Horizontal. And now choose focus. And choose focus text. Alright, now you can give it a title if you want. Yeah. Yeah, and now you can do it though.
Like, girl. Yeah, what?
Yeah, because you press control.
Oh.
Press control. Now we got all this.
Oh, wow. So you're going to cook it?
Right?
So you're going to buy it?
Once the multi-QC finished, we can remove the deleted one. We can remove the ones that didn't work. Now let's have a look at the output of multi-QC. So click on the eye next to the web page of multi-QC. Alright, and that title that we gave it will appear here. And we can start seeing how everything is combined together. So it tells us about the sample name. We've got the organism and the chloroplast, and there's 1 contig and 92 coding sequences. We can have a look at each one of those reads that we produced, that we gave it. And we can see the GC content here, the percentage of duplication, the GC, the length, And by the way, we were looking at the average length. This is one thing that we were missing from the first table. The average length. We can find it here. 126 versus 6,680. So I can put it here. 126, 6,680. All right. Again, I find it by scrolling right in the general statistics, the length. Now we see the results from PROCA. See the results from PROCA? We got how many coding sequences, how many ribosomal RNA, how many transfer RNAs. You can copy it or you can take a screenshot. And you can paste it under the genome assembly, genome annotation. Right? And then you can write it in the text box if you want. Now the alignment comparison. You can go to the multi-QC report. First of all, FASTP tells us how many reads were filtered and so on. Sequence quality. So it gives us so very similar results to FastQC. So FastP and FastQC provide fairly similar results. We already gone through them. You don't need to have another look at them. Sequence quality histogram, we looked at them separately, but we can look at them here together, side by side, which gives us a good sort of, a good indication how they compare to each other. The length distribution. I didn't put the qualimap once. What did I miss? Yeah, I don't see the qualimap results. All right. So to get the mapping results, we can just look at the output of qualimap of each one of them. Alright, so now I went to the qualimap reports. Alright, click on the eye next to the qualimap reports. For some reason, multi-QC didn't include the qualimap outputs here, so we'll just need to look at them separately. And you can find here the information about how many reads were mapped. And just make sure that you interpret it correctly, that you're looking at the right one. What do you think? That one is the nanopore. All right, there's only 180 sequences that were mapped, and we would have a look at the read length. Minimum of 55, a maximum of 6,000. So it's definitely looking at the nanopore one. Sorry, a maximum of 22,000. So I can put them. And by the way, we didn't use Minimap. We used BWMIM2 as well to map the nanopore ones. The mean coverage, just find it. If you scroll down in that report, you can find the mean coverage. 8.6. Mapping quality. There you go. And the general error rate will be at the bottom. There you go, general error rate, almost 14%. Yeah. Quality reports. Scroll down. I think you can find the mapping rates, coverage will be down there. And I can do the same for the Illumina reads. So if I go for the second Illumina quality map report, all right, the mapped reads. The coverage. The mapping quality. And the error rate, general error rate. So when I look at this table, what's sort of the main difference between the nanopore and the Illumina alignments?
Covers are not.
Alright, so as you can see, the main difference is we can find in the coverage and in the general error rate. So the coverage, first of all, comes from the fact that we only use, at the end of the day, we only use 180 nanopore reads. So the vast majority out of 2,000 reads, a lot of them were dropped because of poor quality. We started from 2,000 nanopore reads and only 179 were used for the actual alignment after the quality check. And the error rate was still much higher, substantially higher than Illumina. But still, the length of the nanopore reads allowed us to have this one continuous assembly. Let's quickly try and go over the last step. The last step is actually visualizing how those reads aligned back to the reference assembly, genome assembly. So if you type JBrowse, JBrowse will launch a genome browser, an interactive genome browser. And we need to tell it what data we want to visualize. So we start with a reference genome. And again, rather than a built-in, choose the genome from history and choose our final assembly. All right, the tool called JBrowse. JBrowse.
Yeah, genome browse.
Yeah, just a single one. Yeah, just a single one. Right. Now, so this will be a reference, and now we can overlay on it tracks with extra information. So let's insert a track group, use the plus sign. And in the track category, let's call it gene annotation. And then we insert an annotation track. And by default, it's going to choose our only gene annotation file that we have in our history, which is the output of PROCA, the GFF file that PROCA produced. So we don't need to change anything in there. There's only one type of file that fits there. Now scroll down, don't worry about all the options until you get to another insert track loop, another plus sign. And click on it and then add alignments. And then let's insert another annotation track and change the track type into BAM pileup. All right.
It's just the process.
It's still, yeah, all right. All right. Yeah. No, choose the fragment and then choose the final assembly. Even though this one will be fine as well, but choose the final assembly. No, just the right?
Yeah. I mean, he said you can work if you want. Or you can put that if you want.
Seems like it's mainly the lab computers that have trouble loading. Yeah. Yeah, we want the GFF features and it automatically will select the GFF file.
Yeah. Yeah.
Alright, and if your browser is not stuck, so choose BAM pileup and choose one of the BWA meme outputs, either the Nanop or the Illumina, and then add another track, annotation track, with the other BAM file. So in first of them choose the nanopore and the second the Illumina, or the other way around. All right, but just make sure you add them both as separate annotation tracks. Yeah, there's only one. Yeah, scroll down. Insert track 2. Yeah, call it alignments. Yep. You can call it alignments, the second track. Matter, and then insert annotation track. Yeah, you can remove that one, yeah. Insert track, and choose change it down. Yes, and then choose, that's right, first one. Now scroll down, insert another annotation track. Change it down again. And choose. No, it should be, yeah, just double check. Yeah, you got the nanopore here, and then click on next. It's alright, scroll down. Scroll down, insert another track group. Always alignments. Alright, sorry, yeah, alright, you did it already. So change that into BAM. Then choose, that's fine, nanopore one. Now scroll down a bit and insert another annotation track. And change it to BAM again. Now choose the second one. Yeah, scroll down, now just the leaves. Yeah, now we can execute.
Prefer window.
Alright, I will just execute, so I hope it will load.
Window. The key is for 330 and master 6 the cost of
Was Alex last week?
I'd say it's
no,
supposed to have
he's 4 and 5. No, sorry, 5 and 6.
So you're supposed to have two track groups. The first one has the GFF file. The other one has two annotation track in it, one for each band file. Now, I just want to say one thing. While this is loading, the good thing about Galaxy, so everything is going to be saved. All right. So whether you close your browser, you go home, the next time you log in back into your Galaxy account, you will have the same history. So you can go back to it whenever you want. You can replicate. You can do any step again. All right. It stays there, and it's not going to get deleted for at least a year. All right. So that's as well one of the advantages of using this interface, that everything is saved under your account. And I'll take the opportunity as well to ask everyone, please, 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 to file in your student experience of course and teaching survey. If you're not sure how to do it, I don't know how to do it either because I'm not a student. But I can tell you that you can go to Experience at Griffith and there's information there for staff and information for students. I've read the information for staff. I advise you to read the information to students. What I can say that there are cash prizes that are randomly given to people who fill the survey. There's $25,000 that are being split to I don't know how many students, but I assume each student gets $100 maybe. I don't know. Has anyone ever won? I don't know. I'm not a student, so I don't know. But there is $25,000 that's being given randomly to people who fill in their survey. So please fill in the survey and say good things. Well, if you say good things, there's a high chance that you get the prize money. I think so. But if you're not sure, there is videos here, how to access the system and so on. Again, I'm not sure what it is for. There you go, winners. You can see the prize winners and so on. And I think there are, if you go, there's what students need to know. So there's here how you need to know and how you access Doe and what is the terms and conditions of the prize. Every course survey you complete enters you into a draw to win a share of cash prize pool of $10,000 in trimester 1 and 2 and $5,000 in trimester 3. So it's $10,000 per trimester.
I think usually at the end of the trimester, they will send an email to us.
Often at the end of trimester it's too late. So that's why, no, seriously, we always wait until the end and the service closed. So, but yeah, you should get an email, I'm pretty sure.
Yeah.
I'll double check that exactly. But just keep that one in mind and please fill the survey. Because if you say good things about us, we get to keep teaching the courses and we get to keep our jobs and things like that. I don't know if you've seen as well. Final assessment. All right, final assessment. We've put in, if you go to the course website, and At the very top, you've got the assessment overview, and you can go into the final assessment. We've opened up the instructions to the final assessment, and we provided an updated example, so you can have a look how a good report should look like, and you can find the details in that Word document about the final assignment. All right, basically you'll have 4 main tasks that follow most of the jobs, most of the tasks that we've done in the course so far, and you'll need to do them for a gene that you choose, but then don't write them as task 1, that and that, task 2. It needs to be written as a report. So again, that's why we provided the example, so you know what we're looking for. It needs to be as a report with references, and it doesn't need to be in the order of the tasks that we're given, all right? Just make it flow nicely, read nicely as a report, and then you just need to include all those tasks. Master students, you will need, in addition to the written report, you'll need to record a presentation of yourself talking for 5 minutes about the gene that you've chosen. All right, so a little bit of introduction, a little bit of your approaches and the results. So pretty much summarizing the report in 5 minutes, 5 slides. I will still, for the masters, there's no, just submit it together. Just submit together with your written assignment. So you submit the document and the recorded presentation in the same place. As I said, there's a Word document with very specific instructions. If you're not sure about anything, let us know.
Yeah.
Alright? Let's just check. There you go. Just before we finish, I want to show you the output of JBrowse. So if you click on the eye next to JBrowse, it will open an interactive genome browser. And if you entered all those tracks, all the tracks should be available on the left-hand side.
Is it due on the 15th of June?
I think, or 11. I think 11. Yeah. At the moment, the tracks are not available, but they are not visible. Sorry. But if you tick box, so if I'll start taking the gene annotation, and suddenly you'll see that there are gene predicted along the genome. Now, when you zoom in, if you zoom in into that, you can get all the way down to the sequence level. All right, and these are just represented as colors, but if you zoom in a bit further, you'll actually see the nucleotides. And we've got the sense, like our genome assembly will be the colored one, the sense and the reverse strands. And on top of those, I would ignore them. These are all the possible amino acid translations. Like the 3, they're a bit cluttering. I would remove them if I can. Not sure if I can. But the interesting bit then is to scroll. You can scroll right and left and find predicted genes. So what I want you to do is have a look at the chloroplast, or the tobacco chloroplast genome. Have a look at the genes you found in there, or just scroll around this and just choose a random gene. For instance, in this case, tRNA of arginine, and I can click on it. And you can get the information for that particular gene that's predicted. And that's its position. So if I want to, if we say, well, I've chosen that gene, this is the position of the gene. You've got the sequence and all sorts of information that comes out of it from that annotation. Now the interesting bit is when we overlay the alignment files. So if you tick box those both alignment tracks, we can see each colored bar represents a read from our raw data. And the color represents the alignment, whether it was put forward or whether it was flipped and put on it. And have a look at these, what we see here. What do you think? Which data is that? Is it the Illumina data or the nanopore data?
Short reads, the blue ones are.
These are the short reads. These are the Illumina data. You see they're all the same size, the same length. And there's a lot of errors in there. So everywhere that there's a colored bar, it represents that it's different from our assembly, from our reference. All right, so if there are not too many colored bars, it means that there's not too many errors or too many other bases compared to the reference. And that's basically how the assembly was done. It was looking at each position and it looking what's the consensus. So everything that's non-colored is the consensus, and that will be the letter that was determined, the nucleotide that was determined. If I scroll down and have a look at the nanopore data for exactly the same regions, it's a bit hard to see, but first of all, the coverage of nanopore. You can see that just long reads that cover the entire region. All right, there's no gaps in between them as opposed to the Illumina. But they've got a lot more sequencing errors scattered within them. You can see many, many, many different colored bars, and each one of those represents a sequence error, which I can, from what we saw from the quality check, it's understandable, but that gives you a good sort of view to see. So what I want you to do is just focus on the gene, say this is the gene, that's its coordinates, and then take a screenshot just to represent the difference in, for instance, look at this region. All right. Again, overall, I've got, there's gaps, but there's a fair bit of overlap between the Illumina reads. But I assume if you scroll right and left across the genome, you'll be able to find a region when there's major gaps in the Illumina reads, where the Illumina reads do not cover and the nanopore does cover, or the other way around. All right, so I just encourage you to go and browse here and just find region that looks interesting. You can zoom out, so it will be easier to identify those regions. And it just gives you a sort of understanding of how, see the Illumina reads coverage versus the nanopore reads coverage. So actually the nanopore one is limited. There's some region that nanopore doesn't cover at all at the beginning. All right, and some regions with less Illumina coverage. Yeah, I must say the Illumina coverage is not bad at all. All right, that's pretty much it. Took us a bit longer than I expected, but we made it to the end. It's still running. It's fine if JBrowse is still running for you. As I said, it's fine. You can close the computers. It will still run. You can log in next time or later into Galaxy, and it will show it. All right, it's not lost. Thank you. Yes, just like that. So you see the coverage in Illumina is much more substantial, like, you know, you get an average. And you can see it from the table, in average you get like 50 reads cover some region, whereas in Illumina, or in Nanopore, you got an average of 5 or, you know, 6 to 8 reads. And you can see it in the coverage, the mean coverage, so that's sort of that. Then when we look at the visuals, we can see that. Yeah, and if you zoom in a bit, well, you can still see, so you see all the bars, all the colored bars there, they're all errors. And if you zoom in a bit more, it will be more visible. Yeah, there you go. So you see all these colored bars represent sequencing errors, whereas in Illumina, the majority of them is very consistent. There are some randomly scattered, but not as much. And again, it's the same thing we saw from the statistics.
Like explaining it again, I'm a bit lost. But when it comes to this, like, well, what are we looking at? Is it the color? The color explains the difference, or
The color of the bar,
you mean these different type of colors?
yeah, they represent different nucleotide from what the reference has. That's why they're different color, because it sometimes can be, you know, it's A instead of a B, or A instead of a C, or A, G, C, or T. So each one of them will get different color.
Okay, and when it comes to the space in between this white region, which means not in the reference.
No, it doesn't mean it's not in the reference. It just means it aligns. You know, there's not a full, but overall, like, there is a full overlap. It just lays them out like that. But overall, you do have a full coverage. You know, if you go take a position and you scroll down, there's, you know, you've got multiple reads covering a position. But they're not, you know, they're not continuous. Because they're short reads, you know, they're aligned like that.
So are you saying that those that is the white color means this?
So the white colors, these are gaps. So there's nothing. So this is a read here, and there's a read here. All right?
Mm-hmm.
And in between them, there's no, well, in this case there isn't, but like this read covered that gap.
You mean
The one on top, the red one, yeah.
So every single line is it?
Every single line is a read.
It's a read.
And the read is matching that point of the reference. That's what the alignment is doing. It's taking all the reads and it finds where they align on the reference, and it positions them in the right position. And then it just sort of layers them in such a way to help us visualize it. But, you know, it could have been just randomly, like they could have been one below the other or something like that. It's just a way of visualizing it to represent it a bit less messy.
But it seems that Illumina has a lot more as compared to
Coverage,
Nanopore.
exactly. So that's what I was saying. That represents, for Illumina, in average, you got 50 reads covering a position.
Yep.
And
yep.
in Nanopore, you got an average, just 8 reads covering a position.
And that is the one that will fill up the table on the top. Which
Yeah,
is the
well, which comparing the alignment rates. All right.
Yeah. So you want us to take a photo of?
Yeah, I can take a screenshot, you know, and show, yeah, and that represents those differences, both in the sequencing, like in the error rates, and also in the coverage.
And do you need us to take this, or do I zoom in further to this?
No, I wouldn't zoom in more. I might even zoom out. If you zoom out, you'll see more. Or if you do it on a large screen, you'll be able to see more Illumina reads, so you get more.
Right. Okay.
But yeah, no, that's fine. You know, but if you, and if you're in the region that shows the gene, that's fine. So it's not a lot. It's the one that before it was mine. So it needs to be aligned already. So how about try it? Choose the performance one. So that one is already Alright, it needs to be already aligned. Alright, and if it doesn't show the right taxonomy, will change the name of the aligned ones.
She's like, why they're not doing? Why they taking so time? She's like, even we were busy with it. आज वो घूमने गए.